We can. Yeah.
Yeah.
Yes.
So, sir, this presentation, how much time you want, sir?
Sorry?
Time, how much time you want, sir, for this
I think
presentation?
around 45 minutes.
45 minutes. And then we'll have some interactive session also. So
Sure,
that
no problem.
our these informations are there, and the PhD scholars are also there, and
Okay,
so it will be very useful for them to interact
sure,
with you all.
sure. Thank you very much. Yeah.
So, Dr. Jovit, shall we start now?
One and all present here. Yes, sir. We'll be starting now. So, very good afternoon to one and all present here. So, respected Dr. Giriraj Chandak sir, our speaker for resource person for today's webinar, respected HOD sir, and all my colleagues and beloved scholars and students. So it's my privilege, and I'm very happy to host this webinar hosted by Department of Pharmaceutical Chemistry as well as IIIC, JSS College of Pharmacy. So now I request Dr. Afzal Assam, Head Department of Pharmaceutical Chemistry, to give introductory remarks of this webinar.
Yeah.
Please, sir, over to you, sir.
Very good afternoon to all. And it is my pleasure to welcome you all for this session. This session, I think it is very useful, and this is the starting point for your research work and all work, because genes is the main. Nowadays, COVID situation also, you see, they are targeting genes. So this gene, you should know, all the PhD scholar and the students, even the faculty also, they should know about the genes, what is the genes, and how we can manipulate the genes, and how we can target the genes. If you know about the genes and the genes mutations, then it is very, very useful for you for the future work, future endeavors. All. So for this, we have invited Dr. Chandak. He's a group leader, CCMB Hyderabad, and he's having a lot of experience in this field. And I think this presentation is going to be a very useful and very useful to all of us. and I suggest all my research scholars and faculty and students to use this for the future their work. So with this, I will not take more time because sir is waiting for the presentation. So I request Dr. Jubi to just introduce Dr. C.R. Chandak. Over to you, Jubi.
Privilege to introduce our webinar speaker, Dr. Ratan Chandak. SAR is a chief scientist and group leader, and presently is affiliated to the Center for Cellular and Molecular Biology, that is CCMB Hyderabad. His specific interests are molecular diagnosis of commonly occurring rare diseases, polygenic score-based genetic testing for complex diseases, genetic and epigenetic basis of complex disorders like type 2 diabetes and related intermediate triad, with the focus on fetal origins of adult diseases. To his credit, SAR has got many prestigious awards and honors like SAR's Fellow National Academy of Sciences, Fellow National Academy of Medical Sciences, again Fellow Telangana Academy of Sciences. He is recipient of several Oration Awards. He has published more than 100 papers in peer-reviewed journals, including Nature, Nature Genetics, and Lancet, etc. So we are so blessed to have with us today. So with this brief introduction, may I request Dr. Giriraj Ratan Chandak to start his presentation. So over to you, sir.
Okay, thank you, Dr. Afzal, Dr. Jubi, and welcome to this webinar from my side to all the students, scholars, professors, anybody who is there. So when I was actually asked to talk about something related to genetics, I thought I'll try to keep it very simple to begin with, and then take on to something a little more complex. So what I have done is I have decided to explain you all this as per my journey through this field. Can you all hear me?
Yes, sir. Yes, very well, sir. Very well, sir.
Okay, fine. So, so as I was saying that I have just tried to build up my presentation with reference to my own journey. When I started my research career in 1995 or 25 years back, when everything was pretty simple, and today we are in a very complex situation, particularly with COVID coming up, the life is really very, very complex. So I'll try to share my story, and through that, I will show you some of my published work and some others which I am taking example for others' published work to drive the point that there are genetic disorders and they can be very simple to they can be very complex. And as the knowledge advances, even a simple genetic disorder can be turned into complex disorder. And that is all because of the possibility of high quality research in every part of the world. So, as I was telling you that I graduated about 30 years back and I became sort of a simple medical graduate. And now, after which I moved to CCMB about 25 years back. And today I have almost forgotten all my medicines. I don't know how to practice. I'm a forgotten clinician. And as I will demonstrate to you that although I claim to be geneticist, but the situation is so confusing that I call myself as confused geneticist. And then when you do such kind of work, you need to understand epidemiology, and then further something which is not explained by genetics, you try to explain by epigenetics, which looks at the factors or the nutrition or alcohol or environment or drugs which can influence the genetic structure. And lastly, if you do all this with my background of medicine, I would like to take this to the community, and that means that the public health is equally important. So I'm sure, friends, that you all are ready to go through this particular journey, and the major focus would always be that whatever research one does keeps the society in the mind and then tries to deliver the fruits of research into the society. So as I said that I started my career as a physician, and during the time 35 years back, I was really running from bed to bed with stethoscope and really giving treatment to the patients. Then I decided that, well, this treatment is just not enough. We need to get into the preventive aspects. We need to understand things better. That is when I moved to CCMB, which is a CSIR institute. And there I was largely taking the things which were identified in the bed with patients to the bench. And then finally, whatever results come out of this, then again take the results identified in the bench to the bed. And that is what exactly I have tried to do. I have taken the knowledge which has been there in the public domain, use that to give some benefit to the society, and then done some good quality research and then tried to put it back to the society. So for any such journey, friends, you know that you need a wonderful team, and this is one of the recent pictures of my team. These are the colleagues who actually allow me to stand in front of you and talk about these things which I'm going to do now. Similarly, when I have done all my research, I have been privileged to have large number of collaborators, because if gone are the days when you can do research in sitting in your room or one corner. You need to collaborate, you need to find people from all over the world. And I'm thankful to all these people to have allowed me to do good research. And these are some of my collaborators who have helped me do good quality research. So let's come to the paradigm 25 years back. When I started, it was said that all diseases may not be genetic. But now after 25, 30 years, we know that, well, all diseases are genetic. Only thing is that the time or the pattern or the nature of disease may be different. Like say we know that there are single gene disorders like beta thalassemia. I hope everybody knows about the gene, but I'll just give you a very brief example just after this slide. So there is a beta globin gene because of which the beta thalassemia can occur. Then there are diseases like Alzheimer's, where many genes can be mutated, because of which the polygenic disorders occur. Then there are complex disorders like diabetes or asthma, which means that there are both genetics that is involved as well as the environment in terms of your lifestyle or the diet or the family history, that what determines the prevalence of these diseases. And lastly, even you know that infectious diseases are also known to be genetic, as Dr. Afzal mentioned that even for corona also we are trying to look at that, why the disease is affecting and so severe in other parts of the country but not in India. Is it because of a different genetic structure of the Indians? So just to bring you to this very, very basic slide, this is in fact the structure of a nucleus. And as you would see that in the nucleus, there is a 1 meter long DNA, which is just coiled around itself and finally packaged inside this 10 micron nucleus. The DNA is a double-stranded DNA molecule. It's held together because of these hydrogen bondings. And as you would notice that adenine with thymine and guanine with cytosine. So this is the structure of the DNA. I have put this much as an example. You know that the central dogma of molecular biology suggests that taking the structure of the DNA by a process of transcription, the mRNA is produced, and then by a process of translation, a protein is produced. And genetic code being triplet in nature, it gives specific amino acids, and it is the primary sequence of the amino acids in a protein which determines its secondary, tertiary, quaternary structure, and finally the function. So if I have to translate this central dogma in little simpler term, then the gene is a part of the DNA, is a piece of DNA which codes for at least one protein, which means that each gene will code for at least one protein. And we have about 30,000 genes which code for at least one protein. So we all are combinedly mixtures of all these hundreds and thousands of proteins. You would notice that although there are similar genes, similar proteins, but each one of individual are very, very different. And that is what is the basis of genetic basis of diseases. Now, what can go wrong? You know, as I said that everybody is looking at genes, there are similar genes. So what goes wrong? So what can go wrong is that your DNA can be lost, it can be gained, or there can be incorrect spelling. If you might recollect that I showed you a sequence, and it is that DNA sequence which determines the protein structure. So if there is any abnormality where there is a particular base is lost or gained, which means there is a deletion or there is a duplication or there is a substitution, which means that instead of adenine, say there is guanine. So all these things can lead to change in the amino acid. Similarly, there can be genes which can be lost because of chemical or radiological damage, or say chromosomes can be lost or gained. We know about Down syndrome, Turner syndrome, where chromosomes are gained or lost. Now, whatever may go wrong, major thing is that there is a change in the information because of which the proteins that are produced, they also become changed. And as I mentioned that it's the proteins which determine specific function. That's why in the absence of an abnormal protein, a disease can be produced. I just want to introduce these 2-3 terms before I proceed, because these are going to be the central terms. Now, as I showed you that there are 2 copies of the chromosomes, and each of these, they are known as alternative alleles. Now this position on a chromosome, this is known as the locus, and A and G as an example are the alternative alleles. So each of us have 2 alleles of a particular locus, and The genotype, which shows both the alleles as a combination, is what is represented here. So an individual can be AA, that means on both the copies it has AA. This is called homozygote for A allele, this is homozygote for G allele, and this is a heterozygous or a carrier. Now let us come to monogenic genetic disorders. What do you mean by monogenic genetic disorders? Now here is the cartoon. This essentially means that there is only one gene which is responsible for causing the disease. And there is a specific inheritance pattern as per Mendel's laws. And that is why the inheritance can be dominant or can be recessive. Now, because there is one gene and inheritance pattern is fixed, that means that the impact of this gene in the population will be fixed. And that would mean that if I carry the gene, then I am affected. If I don't carry the gene, I am unaffected. And lastly, because of all these above three factors, the genetic risk in different families will be similar, which means that whether a family is from India or from US or from South Africa, if this is a monogenic disease and carrying the same mutation, the disease will be similar. So this is what I was, friend, 25 years back when I started my career. And now we will move towards to take my journey forward. Now, whenever such situations are there, as I said that as a physician scientist, my idea was to take this benefit to the society. Now, if I talk about a specific disease, and if that is a genetic disease, I first need to establish. So I need to find out the clinical and the family history. After taking the family history, we understand whether this is a genetic disease or not. Then we need to establish the clinical diagnosis by doing some additional tests. Like say you go to a clinic and you say that, well, I'm having fever or something. The doctor immediately advises you to get a blood picture done. Based on that, they diagnose whether it is viral fever, bacterial fever, and other things. Similarly, focusing on the diagnosis, we then move forward to establish genetic defect. I'm not going to go into the techniques of genetic defect identification, at least for today, but take it from me that there are n number of techniques by which a simple one base to every base in the genome can be detected. Now, once the genetic defect is identified, we screen for the carrier status in the family, and then we find out that who are the carriers, and then we advise a counseling during the time when lady gets pregnant, and we do the prenatal diagnosis, which means that we collect the sample from the womb during the pregnancy and then identify whether the baby in the womb is affected or not. And particularly because we have established the genetic defect, that's why we can with confidence say whether the baby will be affected or not. So here is the example of a clinical and family history. The disease is sickle cell anemia, which is an autosomal recessive disease. I don't know whether you read this or not, but let me explain that this rectangles are males and circles are females. And the red color indicates affected individual, whereas yellow color indicates normal individual. And wherever there is a half circle, that would mean they are carriers. So autosomal recessive disease means both the alleles on that chromosome are defective. And that is why you would find that these two individuals are affected. Now, because these individuals are affected, that means their parents must be carrying one defective allele, and that is why they are carriers. You would see that in autosomal recessive disease, individuals can be affected or unaffected because of the pattern of Mendelian inheritance. And that would mean that we need to figure out by a family history whether which individuals are at risk of getting affected. Now, what is the cause of sickle cell anemia? I showed you the sequence. So if in the sequence at any point A is replaced by T in this particular codon of beta globin G, then what happens that instead of glutamic acid in the hemoglobin A, it is replaced by valine, and that is why there is a sticky patch which is generated on the surface of RBC, and from a biconcave RBC, this become a sickle shaped whenever they are exposed to oxygen tension. Now you may ask me that, well, it is so simple to diagnose sickle cell disease. Why bother about genetics? But you cannot identify this very early on, because these patients, only when they present with some symptoms, then only you will be able to diagnose. But by genetic testing, it can be done much earlier. Now, after clinical and family history, we do additional tests. So these are the biochemical tests. You do a paper electrophoresis where you load the hemoglobin lysate. And as you would see that here, a normal individual shows two bands, an adult band and a normal band. Hemoglobin S shows a big S band, and a heterozygous shows both a S band as well as an A band, which means that this diagnosis is further substantiated by a biochemical pattern as well. And then we move on to the genetic detection. I'm not going to take too much time on this. Essentially, this is a PCR based genetic detection where you would see that A and T, these two will be digested differently by different restriction enzyme. Hence, there will be difference in the length of these fragments, and that will be detected by gel electrophoresis. And again, I'll not go into detail, but you would see that the normal individual, which is this one, it's very different from an affected individual like this or the carrier. Just by the number of bands, you can say the guy having 2 bands is normal, the guy having 3 bands is a carrier, and the guy having 1 band is actually affected. So the point I am trying to make till now is that it is possible by clinical diagnosis, by genetic test, to identify mutation for a particular disease. And you can identify who in the family is carrying the disease and who is not. Once you have done that, then you do a pre-pregnancy counseling, because you want to explain to the family that there is an option of prenatal diagnosis, which means that whenever the lady gets pregnant, there is a possibility of a sample to be pulled out, and this sample can be tested for the specific genetic defect. So these are, the different stages, like 8 to 12 weeks during pregnancy, chorion villus sample can be taken. At 14 to 18 weeks, amniotic fluid sample can be taken. There are many techniques by which the fetal sample can be collected through ultrasound. So I'm not going into detail. Suffice it to say that now with advanced technology, it is possible to collect the fetal material while the baby is in the womb. Now, because this is all done by a blind procedure, so what we do, we take the tissue out and then we tease out the maternal tissue. Then we do an isolation of the DNA, check the sex of the fetus, and because we already know the mutation that is responsible for the disease in the family, we perform exactly just 1 or 2 experiments, because the DNA that is isolated from these samples is very minimum. And finally, because this is a blind procedure, that's why we check for maternal contamination. And friends, last but not the least is that because this test is going to determine the life of an individual, we get it tested by having the experiment by another individual. So with this experiment, we can exactly find out whether the baby is going to be affected or not. And so the affected babies are selectively terminated, and that is how the genetic burden of these diseases on the society is prevented. Now, what do you do after this? Suppose there are different kinds of inherited diseases. So there are dominant disorder, where even if there is 1 copy of the chromosome is defective, it can cause the disease. So comes the concept of genetic counseling, which means that if we know that a particular disease is dominant in nature, by family charting, we can say that it's only a 50-50 chance that the baby will be affected. And if this is a recessive disorder, that means the baby needs to have both the defective alleles, then there will be only 25% chance. And for X-linked recessive disease, which means that it's one of the X chromosome is defective, so it doesn't affect the females, but only males are affected, and hence there is a possibility that 25% males will be affected and 25% of the females will be affected. Now, I just want to point out that these are the possibilities for each pregnancy, and based on these possibilities, the genetic counseling is done. So as I said that you do genetic testing, you now have positives, negatives, and then you counsel the patient and the family, tell them about what will be the nature of the disorder, how the disease will develop, how the disease will get transmitted, what options are there to prevent the disease or what treatment could be possible. And finally, you have to advise the parents that they should accept the disease early and go for an early training. Now, so to summarize this much part of my talk, the points to remember are that for any genetic disease, diagnosis, prevention of further transmission, you need to have a detailed clinical and family history. You need to establish a diagnosis, then identify genetic defect which causes the disease, and then screen for the carriers in the family, do a counseling to them, tell them that what is the risk that they are of developing the disease in the next generation, and then finally help the family by prenatal diagnosis and genetic counseling. So for last 20 years at CCMB, we have been doing this, and I have been in charge of this facility. We have done close to 25,000 patients for genetic testing. We are testing about 35 disorders. We have done about 3,500 prenatal diagnoses, 40,000 families for genetic counseling. And at the same time, we have interacted with various NGOs and patient support groups like musculopathy, hemoglobinopathy, and we have helped them develop the capability of doing these tests. So, which is very satisfying for a simple genetic disorder because of the simplicity, because of one gene and very few mutations being responsible for the disease. So I gave you the example of sickle cell anemia, and it was very satisfying because sickle cell anemia is globally caused by just one mutation. Whether it is India or South Africa or USA, it is always caused by one specific mutation, that A to T change, which means if you have any simple infrastructure, you can actually do this testing, and the disease occurs very early on. But when we had an experience of large number of such patients, we found that actually there is a variable clinical severity in these patients. And what happens is that there some patients present very early, some patients present late, some patients are presenting with lots of complications, certain others never get complication, some people die very early, and some people can live up to 40, 50, 60 years of age. And when we started investigating, because it's very surprising that there is only one mutation which causes the disease, and as I showed you that it is the genetic change which leads to amino acid change and the protein structure difference, it was very surprising that this can lead to a different kind of clinical pictures. So we investigated, and this is the research that we did about 20 years back. We found that other factors like alpha thalassemia there can also be there. And then most interesting finding was that if an individual has high fetal hemoglobin level, then the individual has lower clinical severity. And then we found out that the fetal hemoglobin and the sickle hemoglobin, they interact with each other, and hence there we started looking at the genetic regulation of fetal hemoglobin. One more important part was that there are various drugs which are used for sickle cell anemia treatment. One of them is hydroxyurea, and hydroxyurea treats or manages the disease by increasing the levels of fetal hemoglobin. So we thought that if we can identify some genetic variant which influences fetal hemoglobin level, that will be extremely interesting. So what we did, we took the samples from different tribes where there was very high fetal hemoglobin level, and we tried to look at a number of mutations in the beta globin gene. Again, don't go into the details of this, just see that this minus and plus, they indicate the two states of two alleles. And as you would notice that these are the 5 haplotypes on which the sickle cell can occur. This Benin, Bantu, Cameroon, they are all South African phenotypes, whereas Arab Indian and Senegal are largely Indian. Now, if you see this particular polymorphism or mutation, you will find that it is all over minus, minus, minus. But then in the Arab Indian, this is plus, which means that Indians in particular carry this particular mutation, which lets them have high fetal hemoglobin level, and that is why the disease severity is very low. And that is something which we published in I think 99. And since then we thought that we have won the war and we understand that how fetal hemoglobin is regulated. Now since then, actually we provided the first indication that genetic modifiers may play a role in monogenic diseases. But we didn't realize that this was getting us into the path towards complexity. Now 20 years have passed, friends, and we now know that at that time we identified only one gene. Now we know that there are 38 genes which regulate the fetal hemoglobin levels. And each of these genes have their own effect, but together there are only 3 genes which account for 20% variation, which means that if I am carrying a specific mutation in these 3 genes compared to you, still only 20% variation of the fetal hemoglobin level will be explained. And these mutations or polymorphism, they are better known as now, they are known to be associated various complications of sickle cell anemia. So the point I'm trying to make is that although sickle cell anemia has one mutation which causes the disease, there are many other genes or genetic variants which can modify the clinical course of the disease, partly by regulating fetal hemoglobin level, and we are currently investigating what else they can influence. So what this led us to was to figure out that, okay, what is monogenic disorder and what's a complex disorder? As I told you, monogenic disorder is because of one gene, one inheritance pattern, risk in families and risk across the world will be essentially similar. But in contrast, if you take complex disorder means that there are many genes which will be finally deciding the clinical picture. And each of these genes will have its own pattern of inheritance. And that is why this will be a complex inheritance pattern. Now, as I mentioned that the clinical picture in a complex disease is influenced by environment and lifestyle. That is why the genetic risk will also be very different in different population, depending on the type of genetic risk and the type of environment and lifestyle used in the population. So you see that how different it is from single gene disorder. You can just identify gene mutation and you can help the whole family. But here you have to look at so many genes, then you have to figure out how many environmental factors are there, and that's how only you can counsel. But unfortunately, all the single gene disorders are rare. It is complex diseases like diabetes, cardiovascular disease, these are very common. And that is why we started working on complex disease like type 2 diabetes. But what I'm going to demonstrate is that we did not leave the simple disease pattern also, and that's how we could make a difference. So as I told you, friends, I started like this 25 years back, and today in the complex disease era, now I am this. So so the point I'm trying to make is that it takes a long time. You have to be patient in research, and if you continue with patience, with well-developed ideas based on the earlier work, it's clearly possible to achieve what you want to do. Now, when I said about sickle cell anemia, because we were able to work on so many patients and so many individuals, we realized that there are many lacunae and limitations. So many limitations and lacunations that we decided to have a mission mode project. We came to know that in a particular area, particular states, there are high prevalence of sickle cell anemia, like Chhattisgarh, like Jharkhand, like Odisha. So we put up a mission mode project where the mission was that we will manage the genetic burden. We will understand that why certain individuals are responding differently to different drugs. We also try to discover some drugs using, hello? We also try to discover some new drugs. Then We also wanted to do a treatment for these patients, so the gene editing or CRISPR based approach. And finally, we wanted a very simple point of care diagnostic kit. And as all you know that these kind of work can only be done when there are different institutes collaborating. So we were 7 CSR institutes, 6 hospitals, and together we started a screening program. Again, I'll not go into the details of the whole thing, but we undertook to attend the complexity in the phenotype of a disease which is caused by one single mutation. And what we did, we went to various schools, colleges, and different type of screening we did. Suffice it to say that finally we were able to identify who are affected and who are carriers. We also organized various camps, and that's the way we could identify clinical details, family history, and now we then embarked onto the study of the genetic basis, which is almost similar to what I mentioned to you about single gene disorders. And what we could achieve in this particular project, what we could achieve was we have now screened 20 lakh children. So one of the lacunae was that what is the burden of the disease? We now know that this is 10% carrier and this is only 0.4% is the total patient. But if you realize that in a population of 130 crores, if you have 0.4% patient, that leads to lakhs and crores of patients. We've identified various compounds. We have a point of care diagnostic test. We have generated these iPSCs, which are currently the known ways that the disease can be treated, and we have engaged with government hospitals, NGOs, and patient support groups, and we have now moved on to various other states also. So what I try to say that you start with a single gene disorder, but if you are focused and if you have the desire to push, you can actually lead to understand the complexity of the disease and finally help the patient. I will take a couple of minutes more to take the example of complex disorders, particularly type 2 diabetes. And the implication of this is because it's about 15 to 20% of the population that is diabetic. So if you take an etiology of a complex disorder, suppose if I take, for example, cardiovascular disorder, then the final clinical picture is a combination of the environmental risk factors or protective factors and genetic risk factor or genetic protective factors. These all interaction determines whether the disease will be early age of onset, whether it will be severe, or what will be the clinical symptoms. And as I mentioned that the diet, exercise, lifestyle, they are the environmental factors which can influence. Now, if the complexity comes, because whenever you are looking at these intermediate traits like triglycerides, HDL, blood pressure, which determine a cardiovascular disease, you will find that there are a number of genes which influence each of them. Like HDL is influenced by at least 4 or 5 genes. Triglyceride is affected by 3 genes. So the complexity is that you have n number of genes, you have n number of environmental factors, and it is a interaction between them which will allow you to help the patient. Now clearly the genetic risk cannot be modified, particularly when it is present throughout the genome, and that is why we first determine the genetic risk by looking at all these genes, and then we modify the environmental risk factors, like we subject to them to exercise or to lifestyle. Now you may ask me that what is the point of doing genetic testing, because we all know that a healthy diet, a good exercise, and managing good lifestyle without addiction will have a good life quality. But then you also know that for how long you can actually follow this particular thing. So what we do by genetics is identify the specific individuals who are at risk and then substitute their treatment with these lifestyle modifications. You know that diabetes is a global emergency, and it is also known that it's a genetic disorder. There are various ways. In India, there are about 15 to 20% population which are affected. Now you may ask me that, well, there is so much research going on diabetes in the whole world. What's the point in doing in Indians? Well, this is an example to indicate to you, as I showed with sickle cell anemia also, that the clinical picture is very different. For type 2 diabetes also, the age is an early disease. The The individuals are not obese, yet they develop the disease, and they are strongly insulin resistant. And hence what we thought that if we can look at the genetics, we should be able to explain the whole of this. And this is what brought us to this particular picture, that if you want to do any complex disease research, you want to have the patient very well identified, and then do all kind of genetic studies, functional studies, everything, look at the interaction with the environment, but somewhere you have to take it back to the patient. Now with this thing in our mind, what we did, we started a long drawn study. I will not go into the details because this is a 20-year study. If you see diabetes in young adults, suppose if I as an individual, I'm a 15 to 30 years old individual, then there can be various different ages of diagnosis. So for individuals with type 1 diabetes, the peak is usually between 10 to 15 years. But if you take type 2 diabetes, this is usually between 35 to 45 years. And then thirdly, if you have anybody with maturity onset diabetes of the young, then their age is also between 10 to 15 years. So what I'm trying to say is that it is not easy to diagnose diabetes just based on the clinical symptom or age of the presentation. There can be all three types of diabetes which can be there. You all are aware that the treatment of type 1, type 2 and MODY is very different. Hence, it is important for us to understand that how to differentiate between these individuals. What I'll do, I'll take example of MODY, which is maturity onset diabetes in the young. It's an autosomal dominant disease, which means if there is only one copy of an allele defective, it can cause the disease. So what we looked at, that in the maturity onset diabetes of the young, it occurs less than 25 years. It's an autosomal dominant inheritance, and there is a defect in beta cell function. When we investigated large number of MODY, we found that MODY can also be of many other subtypes. And that subtype can be because of the specific genes in which the mutations occur. So MODY itself is a part of diabetes, and in MODY there are further subtypes if we do specific genetic studies. Now, about 14% have mutations in glucokinase, then 75% have different transcription factors like HNF1 alpha, 4 alpha, and all these have different clinical picture. So the question was that, well, MODY itself is a common clinical entity. Why should we discriminate between these and look for mutations? Well, this graph is example that why one should do genetic testing for type 2 diabetes, type 1 diabetes and MODY. Now on the x-axis is the age of an individual who is affected with disease, and on the y-axis this is the glucose level. You will notice that a normal individual is born with normal glucose level, and throughout the age of 60 to 80, the glucose levels mildly increase and remain all right. But an individual with glucokinase, by the way, I'm sure you are aware that glucokinase is the first enzyme which converts glucose into glucose 6-phosphate, and this is a rate limiting enzyme. That's why the metabolism is bad right from at birth, and so the baby is born with high glucose levels, but then it continues to have similar kind of high levels, almost a little shade higher than the proportion of normal individuals. But if you see transcription factor mutations, and because these mutations are largely regulatory, they regulate many pathways. So you would find that although the child is born with normal glucose level, but as the age advances, it really shows very severe disease. And as you would see that this blood glucose levels are more than 20 millimoles, which comes to about 350 to 400 milligram%, and as early as 30 to 40 years of age. So clearly, in the MODY itself, it is important to identify the genetic mutation so that their clinical course can be predicted and their treatment can also be modified. Now, can this also have an influence on the treatment? Yes, it can also be there, particularly, as I said, that because glucokinase mutation, it is onset at birth, there is a stable hyperglycemia. So if one can just do a diet treatment, there will be no issue and the individual will be very easily having a good lifestyle. But if you have transcription factor mutations, the individual will be progressively hyperglycemic and they will really have lots of complications, and many of them may need injectable insulin for treatment. Now, does this genetic mutation can also have some implication on the type of drug that is used? So this is some study which our collaborators did. As you would notice that x-axis is the time in minutes of the glucose tolerance test, and here is the plasma glucose level. You would see that this yellow individual is the HNF1 alpha mutation. You would find that the glucose tolerance curve itself at 90 minutes, the peak is very different and glucokinase remains it stably. So if there is a boy, there's a boy with glucokinase mutation presenting as MODY, just safely leave children off the treatment. Just monitor and the children should be fine. Now, we also find that as per the specific mutation also, there can be a difference in the sensitivity to a particular drug. Now, here is an individual whose time since diagnosis is depicted here. So 8 years to 13 years since diagnosis. And this is glycosylated hemoglobin, which you know is a parameter of the control of diabetes. Now you would see that this individual was on glibenclamide, which is a type of drug for treatment of diabetes. The individual was working quite all right. And here, the individual was stopped glibenclamide, and after a wash period, was given metformin. And you would see that metformin couldn't control diabetes, and glycosylated hemoglobin started increasing almost to the double level. And at this stage, again, metformin was stopped and glibenclamide was started. So that means that again started coming back to normal, which means that even in MODY, if there are specific mutations in HNF, I mean transcription factor genes, those are specifically sensitive to sulfonylureas. So the point I tried to show you, friend, was that if you are able to do specific diagnostic testing, it can be used for identifying status of an individual. It can be helpful for deciding complications. It can be helpful for deciding the treatment also. I'll skip this slide because this just tells you how the technological advances have allowed you to move forward. As I said that there is one single variant with which I started with sickle cell anemia, and now it is possible to do whole genome sequencing and identify. And so using all that now, the gap between monogenic and complex disorder is slowly decreasing. Now, what do you do when you look at a complex disease? I think I'll skip this part. So I will just leave it over here. So now, as I said that the type 2 diabetes is a complex disease, you can always argue that why should I get a testing done for type 2 diabetes? Because that is not like MODY. But a recent study which has come in Lancet, it has found out that there are 6 particular variables, the GAD antibodies, age at diagnosis, BMI. These are all, the clinical phenotypic pictures. They are all related to development of complications and this medication. So if we can find the genetic factors which are related to these, then we can help the individual with complications and prescription of medicine. And in fact, there was genetic studies done using whole genome sequencing, and they have been able to divide diabetes patients into 5 subgroups which have different disease progression, risk of diabetic complications, and that will allow further development of tailor-made personalized medicine. So the field is not that bad. You started with 1 SNP, now it's possible to take 53 mutations in one go. And this is example of a cardiovascular disease where if you can analyze 53 SNPs, it's possible to analyze in a chip. And then the individuals who are at risk of developing cardiovascular disease, they can actually be predicted. And so the pattern has come that not for one gene, but you prepare a polygenic risk score, which means that n number of genes are analyzed and then a score is calculated based on those mutations. So the current excitement is that the whole genome is a genetic sphygmomanometer, and an individual's genome can predict many traits, which means that if I am looking at certain SNPs which may be influencing BMI, so as I showed in the earlier one, that if I do this chip-based analysis, there is possibility to identify a number of polymorphisms, and those polymorphisms, if they are related to BMI, and then we know that obesity is associated with risk of type 2 diabetes, breast cancer, and other things. So just doing genetic testing can allow us to predict the risk for all of this. I'll take one more minute to just tell you that the field has now moved to whole genome sequencing, which is now called as next generation sequencing. There are three types. One is whole exome sequencing, where we sequence the whole exome, which means that is only the protein coding part of the genome. It is little over 1% of the genome. But the advantage is that you can identify a disease-causing mutation because these are protein coding regions, and any mutation there will change the amino acid and the protein. So this is really a causal mutation. Then there is targeted sequencing, which means, say, for example, if I know an individual is thalassemia, then I can just sequence beta globin gene and I can actually identify the mutation. And last, it is whole genome sequencing, which means that every base on the genome can be sequenced. It is very extensive throughput and analysis is very complicated. I leave you with two slides. One, which is a PCSK9 story. I'm sure many of you people might be aware, being from pharmacology background. We know that statins are the most effective therapy which is used for cardiovascular events. It lowers LDL cholesterol. But we know that about 20% of patients cannot tolerate the statins. So there was a study which actually identified rare variant in this gene PCSK9, and now they have done various phase 1, 2, 3 trials, and they have found that if inhibition of PCSK9 with monoclonal antibodies can be done, then up to 50 to 60% of LDL level can be lowered. So same thing is possible if you do next generation sequencing. These are two children who had a disease, and because of which they both were presenting with cerebral palsy, but very early on their disease was diagnosed and they were given specific treatment, and that's why at a young age they are as healthy as you and me. So I want to leave you with this slide, that it's a journey from monogenic to complex disorder. It takes long time. You have to be patient, even at the cost of changing your own clinical phenotype. And lastly, whatever I have spoken to you, this has been possible also because I had a wonderful mentor, Dr. Lalji Singh, who was my mentor, was my friend and my guide. So in my journey, there have been large number of individuals who have helped me achieve all this and present all so much of information to you. I hope that you all will be able to take some leaf out of this and build up your own career. Thank you very much, and thanks for your patience.
Thank you so much, sir.
Thank
Of course,
you.
it was a very interesting and informative session. Yeah, definitely it would give a new dimension to us like pharmacy researchers. So now the session is open for question answers. I request the participants kindly unmute your mic and you can interact with our speaker. Please. I request the research scholars and students also. Please unmute your mic
You can
and
ask,
you
you
can
can
ask
interact
the
with the
question.
sir. If you want to ask any question, it is open. Session is open for questioning.
So good afternoon, sir. This is Dr. Rajesh.
Hello.
Yes, sir. This is Dr. Rajesh.
Yeah.
Sir, I have one question. Before that, the presentation was very nice, sir, and interesting. And you have vast experience on genetics, so that's why I'm asking this question. Sir, many
Please.
people, even in literature, I understood that the genetic defects is due to lifestyle modification. How lifestyle modification directly gives impact on these defects? How this lifestyle directly That means the DNA changes or defects due to lifestyle. Right, sir? Is there any direct proportion to the genetic modification? How that happens, sir?
Okay. So that's a very good question, I would say. Can you hear
Yes,
me?
sir.
Yeah, yes, sir.
Okay. So the question you asked was that we have heard that lifestyle can also lead to some changes in the genetic defects, and how that happens, that's what you want
Yes.
to know. So I will first give the simpler answer and then the complex answer. First is that based on your profession, there are possibilities of genetic changes occurring. You know, many of the individuals, they develop cancer because of their exposure to radiation, or people who are working in chemical industry because of specific chemicals exposure. What they do, because of their persistent exposure to these chemicals, they actually lead to certain mutations. We know there is a disease called ataxia telangiectasia, where also because of the specific exposure, the repair cannot take place properly. Similarly, there are different types of cancer which can also occur. So that's the first part. Now, second part is that you have mentioned that how lifestyle can induce some genetic changes. Now, these are not genetic changes actually. These are called epigenetic changes. So I think I mentioned very briefly about that. Genetic changes will largely change the sequence of protein or amino acids so that the protein will not be functional. But epigenetic changes means your genetic structure remains the same, but there are different modifications which take place to the nucleotide base. So there can be some methylation, there can be some acetylation, and that what these epigenetic changes do is they modify the quantity of the RNA and the protein level, which means that although a protein is there, but it is not there at the right time or in the right quantity. And that is what, being regulatory in nature, it can actually cause the specific disease. Now, there is more thrust on these epigenetics because genetic sequence difference doesn't explain everything. And second part is that if there are epigenetic modifications, they are also reversible. And that is what is the current excitement in all the complex diseases, because nobody knew that how lifestyle and other things can reverse the risk. This is said to be one of the mechanisms. Am I clear?
Yes, sir. Okay.
Okay.
Sir, I have another one question, sir. Sorry.
Yes, yes. No, go ahead.
When the patient getting the gene defects by through radiation induced gene defects. So here only autosomal chromosomes get defects. Now this type of defects, definitely from autosomal, there is no inheritance to the next generation. This particular defects will not be transferred, right, sir? But
Yes.
most of the cancers induced for the patients is in the autosomal only. Am I right, sir? So these type of cancers cannot be conducted from one generation to another generation.
Okay. So the question is that because the any exposure like radiation or chemical only induces the change in the somatic cells, and that is why this cannot be transmitted to the next generation. So the question is that why is it that many of the cancers are still found in the next generation? Again, a very good question, I would say. So, Dr. Rajesh, this you are very right that radiation-induced changes will not be transmitted, but there are many cancers which are inherited, which means that these are occurring because of the mutation in the germline itself. So, when the individual is born, that time itself is carrying the mutation, but the defect, it only takes time to manifest at a later stage. If you take breast cancer and the BRCA gene, which is known to be associated, we know that there are specific mutation. So if any lady who is diagnosed with breast cancer, it's the family history is taken, and this kind of genetic testing is prescribed. And I can give example of the famous heroine Angelina Jolie, who actually got bilateral mastectomy done to prevent the occurrence of cancer because she was carrying the risk mutation in her genome. So there are two types of mutations which can occur, and that is why the approach will also be very different. And actually this also brings up whatever I try to say through the clinical counseling, that if you have a clear idea about the clinical history, family history, then it's only you can transmit right kind of information to the patient family. Suppose if somebody has a radiation induced cancer and you put all this fear into all the family members, then you can understand you are creating havoc, right?
Thank you, sir. Thank you very much.
Thank you.
Sir, how is the status of gene therapy in India, sir? How it is
Hello, sir.
successful here?
So, I mean, gene therapy is not very successful anywhere. Most of the studies are, one or two of its kind. But as I tried to mention that now with a collaboration of various CSIR institutes, we have now developed the expertise to pull out the cells, stem cells, transform them into pluripotent stem cells, and then do gene correction and be able to put it back into the cells. We are currently going to do this experiment on specific mice, and after we are successful in that, then we will be moving onwards to the patients. So I would say that a lot of research is happening, but it is still not the stage where it can be used regularly for any patient.
Thank you, sir.
Thank you.
Okay.
Good afternoon, sir.
Yes.
Hello, sir.
Myself, Dr. Gomati, sir. I have one question. What is
Hello.
reverse and forward
Please.
genetics, and how
Your mic is muted.
Dr. Gomati, please unmute your mic. Dr.
Madam,
Gomati,
sir?
unmute your mic. Unmute your mic. Your voice is not audible to us. By the time Dr. Shrikant can ask the question. Shrikant, I think you just wanted to ask something. No?
Madam,
Yeah.
her question is it audible, madam?
Yeah, you are audible. Go ahead.
Yeah, your voice is audible.
Sir, her question is what are reverse genetics and forward genetics, and how we can apply these reverse and forward to revert the mutations? Is there any relation between these?
So forward genetics and reverse genetics, these are, old terms. When people did not have so much of technological advances, that is when this reverse and forward genetics was used. Essentially, what you would do is that from clinical phenotype, you will identify the gene, and then you will be so say, okay, so example of forward genetics is that if you take a patient of hemophilia, which is a bleeding disorder because of factor 8 deficiency, so you know that there is a factor 8 deficiency. That is why you choose factor 8 as the candidate gene and do the genetic studies and identify the mutation. Okay. Now, suppose if there are certain clinical condition where you have no such clinical parameter, there are many such things which are confusing for you. So there what you do is that you take the patients and normal subjects and you compare their whole genome or specific regions of the genome, and then you find out which are the regions which are different between patient and normal. Because you would imagine that, so like say for example, if I am black and you are white, and if I want to identify the gene related to pigmentation, then I compare the genome of mine and yours, and something which is present in you and not in me will tell me that this is the gene associated with this white color. So that is something which is known as reverse genetics, because you are looking at regions first and then slowly narrowing down the gene which may be responsible for the specific phenotype.
Okay, sir. Sir, for infectious diseases, it is very useful, I think, sir.
What?
in case of infectious diseases, so like finding the virulence
Yes.
gene and removing that one. So very useful.
Yeah, I mean, that's what people are. So, I mean, there are many such situations which are there. People sequence, see, because bacterial viral genomes are not that big. So people can sequence that. And after they sequence, they look at the structure, and then they can look at the host and other interactions. They can identify. And that is how they specifically target or design the antibiotics which can use that target. So these are also the approaches that are used. But nobody really, I mean, there are some strategies where people have I mean, very recently there was a paper, I haven't read fully, but what they did was, because malaria is caused by female Anopheles mosquito, so what they did, they mutated the certain genes so that this species became sterile, and then they left them in the atmosphere, which means that if those mosquitoes will mate, then because they are sterile, so there will not be any further production of the future generation. So these are some of the strategies which are used, which will reduce the disease burden.
Okay, sir. Thank you.
Hello, sir.
Yes, sir. Thank you, sir.
Yeah, go ahead.
Hello, sir.
Yes. Yeah.
Am I audible, sir?
Yes. Yes.
My name is Vaishak, sir.
Uh-huh.
Sir, I would like to know, sir, sir, if a fetal is discovered with the sickle cell anemia, means can we do the stem cell therapy like that for the betterment for the disease failure and something like that, sir? Can stem cell therapy can make a good treatment for that child?
Okay. So the question is that can stem cell treatment be done for a sickle cell anemia
Disorder, sir.
child?
Yes, sir. That's genetic disorder.
Or any genetic disorder, right? I'm
Yes,
taking sickle cell anemia because I talked about this. And
Okay.
actually, this sickle cell anemia is the model disease because, as I said, that there is only one mutation. So if one can develop gene therapy protocols, it will help hundreds and thousands of people, right? So I think I briefly mentioned it. What one does is that from the blood of, from the blood, so one takes the blood from these sickle cell patients and then selectively removes out the stem cells. Okay. And those stem cells, they can do a gene editing, put the right gene over there, and then culture that, have enough number, and then transfuse it back to the patient. That is one of the simplest things that is possible. But what people also are doing is that even if you don't have that individual's blood, what people are doing is that take the blood cells from any individual and generate induced pluripotent cells, and then do the correction, and then differentiate that into the hematopoietic stem cell line, and then put that inside the body. So both these things are I would say, not only theoretically, in fact practically possible. It has been done, but as I answered the earlier question to Dr. Afzal, that it is still not in a routine kind of a treatment basis.
Okay. Thank you, sir. Sir, I have one more question, sir.
Go ahead.
Sir, if a child between the age of 10 to 15, sir, if he is getting insulin resistance, means, sir, what would you suggest for a oral medicine, or it's directly insulin?
That's a difficult question because it is not only insulin resistance. There are many other things for a see. First thing will be that if there is somebody showing insulin resistance at 10 to 15 years child, I would really look at the child, whether the child is obese or not, whether there is a family history of diabetes or not. If these two are positive, then my suspicion towards type 1 diabetes will go higher. And so then I will do different kind of testing. So I will do blood sugar, glycosylated hemoglobin, and then I will specifically look at different type of antibodies. I try to show that GAD antibodies, these antibodies will define further for type 1 diabetes. And then one doesn't straight away go to the insulin treatment. It all depends on the type of features or the diagnostic parameters that will come up. But yes, it is said that for type 1 diabetes, because there are autoantibodies to the pancreatic beta cells, that is why large amount of beta cells get destroyed, and hence you have to give an insulin therapy.
Okay, sir. Okay, thank you, sir. Thank you so much, sir.
Okay, it's all right. Yeah.
Sir, this is
Yeah,
Srikanth
go
sir
ahead,
again.
Srikanth.
Sir, regarding neurological disorder,
Mhm.
sir, so which kind of approach we can go through genetics, like Alzheimer's, Parkinson's?
Okay. So,
So,
yeah. So Alzheimer's, Parkinson's, these are all, classical examples of reverse genetics, because there is no biochemical or clinical parameter with which you can diagnose these diseases with certainty. So what people do is that they take people with suspected Parkinson's and largely, normal subjects. How does one go about, because these are complex diseases right from the beginning? Okay, I mean, you realize that people can develop at different ages. There can be phenotypic heterogeneity. Somebody develops dementia very early on, somebody develops later. So these are complex diseases, which means it indicates that there are likely large number of genes which are going to be affecting. Okay, so what one does is that it takes a case control approach. So case means the patients who are very well diagnosed and identified and clinically characterized, and normal subjects who are again equally carefully characterized. And then it can take different approach. Say, for example, if you are suspecting, say, Parkinson's disease, and you have some biochemical parameter, say whatever, maybe serotonin, okay, and you know that there are 10 genes in the serotonin pathway, then you may just take those 10 genes, sequence those 10 genes, and compare between normal and patients. You may be able to find something. And then once you find that there is a particular region, then you can start narrowing it down. Out of 10 genes, which one is affected. But what people do nowadays is that because the technology is available, they look at the whole genome based approach. So they take hundreds and thousands of individuals and then do this kind of chip genotyping, which gives them something like lakhs of these mutations and polymorphisms. And then they compare. Once they compare, they are able to identify certain regions, and then they slowly narrow down to the genes. So this is the way that neurological disease genetics is happening. Of course.
Sir, can we do it college level, sir? This
Sorry?
chip genotyping. Can we perform this one? Start at college level, sir. This chip genotyping, or need.
So these are a bit expensive experiments. Number one,
Okay.
And secondly, anything that you want to do, I would, for a clinical phenotype, you must have a clinical collaborator. And lastly, these kind of studies usually need large number of patients and normal subjects who are very well characterized. Like, as I said, for sickle cell anemia, even if I have one patient, I can identify the mutation. But for diabetes or for this Parkinson's disease, you will need hundreds and thousands of normal and other subjects. And the reason for this is because there are something like 20, 30, 100 genes that are causing the disease. Each of them will have a very small effect. And if you want to detect the small effect, you need it large number of patients so that your study is adequately powered. So you can do at the college level. I mean, there is, as I'm saying, that technological advances make it possible. But only thing is that you need good clinical collaboration, good patient collection, and last but not the least, a lot of money.
Okay, sir. Thank you, sir.
Thank you. Yeah.
Any more questions from the participants? Yeah.
I think.
From my side, a very, very basic question, sir.
Yeah, please.
Yeah, actually, we do design compounds in silico level and go for the synthesis and do the in vitro studies. So my question is how we can relate our in silico studies to the genomics. That's what my question is.
Okay. Yeah. So you decide a target and then design a particular compound against it, right?
Yes, sir. Yes, sir. Yes, sir.
Yeah.
You're
So
right.
so
Absolutely.
that I yeah. So what one does is that whenever you are deciding a target, that means you are very clear about the pathways that are going to be affected when you are designing that compound, right?
Yeah. Yeah. Yes, sir.
Yeah.
Yes, sir.
So since you already know the pathway, it may be possible that the compound that you design may be effective in some individuals, may not be effective in other individuals. So you have a clear-cut path and different
Okay.
enzymes and other molecules known. So those molecules or the enzymes, they can be the substrate for your genetic studies. As I tried to mention about HNF1 alpha or say beta globin gene mutation, even though you may design a particular drug, if there is a mutation downstream on the downstream enzyme in the pathway, then it will not work. So you can first do your structure function studies by docking studies by looking at this compound and the drug target,
Okay.
and then if you find that there are specific residues at which these are interacting, and then you may like to mutate those residues and then see whether you can increase the efficacy or not. Those are the kind of things possible. But again, as we know that this is a very long drawn study, and it really to bring in genetics very early on is difficult.
Yeah. Yeah. Okay. Yeah, sir. I understood. And again, we have to validate our results also,
Yeah.
no, sir.
Yes,
Like
yes, yes.
normally we do the validation studies, but I think it is quite difficult to do the mutations of that particular residues, and again we have to validate
No,
those studies. So can you give some idea?
Hello?
I think Sir has left. I think he will be joining. Sir, I think he has left the meeting. I think he'll be joining
Any other
us.
question from anyone?
I think
You can ask, sir. Sir is having a vast experience in this genetic disorders. So, anyone? Yeah, you can ask. You can ask. His sir is joining now.
sorry.
it's okay, sir. It's okay.
Hello.
Yes, sir.
Yeah, sir. Yeah. You are audible, sir.
It's a
Sorry.
wonderful presentation, sir, and our research scholars and faculty got a lot of information and new dimensions in the knowledge of disease. I think, Garama, you want to ask any question?
Sir, I am asking simple question, sir.
Yeah, ask.
Sir, good afternoon,
Yeah.
sir.
Good afternoon.
Dr. Gaurav, assistant
Yes.
professor, Department of Pharmaceutical, sir. How is genome analysis done, sir?
Sorry, can you repeat?
How is genome analysis done?
Genome analysis.
Yes, sir.
So genome essentially means the DNA of an individual. So if you want to, so again, genome can be analyzed at different levels. As I tried to show that you can look at small gene level, you can look at exon level, you can look at the whole genome level by different types of sequencing. So it also depends on what kind of experiment or what kind of question you are asking, right? Suppose if you want to know the sequence of a particular region because you are interested in understanding what function will it have, then you will do the sequencing and then compare, and that will give you the genomic information.
Okay, sir. Okay, thank you, sir.
Thank you.
Yeah.
Yeah. Okay.
Yeah, I think there are no questions from audience. So Now I request my colleague Dr. Gomati Sanesh to propose vote of thanks. Over to you, Gomati.
Thank you, ma'am. A very good evening to everybody.
Gomati?
Yes, ma'am.
Yeah.
Ma'am, can you hear me?
Yes, yes. Go ahead, go ahead.
Yeah. Yeah.
Thank you, ma'am. Good evening, everybody. As we have come to the closing session of today's webinar on simple to complex genetic disorders, a fascinating journey, it's my pleasure to deliver vote of thanks to all who have helped to make this event a success. On behalf of our institution, I would like to express my deep heartfelt thanks to today's eminent speaker, Dr. Giriraj Ratan Chandak, Chief Scientist and Group Leader, Center for Cellular and Molecular Biology, Hyderabad, for sharing his knowledge on genetic disorders and also spending his precious time with us and along with this interactive session. I would like to express my deep felt thanks to JSS Ahar Mysore and JSS College of Pharmacy Ooty and Institutions Innovation Council for extending their support to conducting this webinar in such a grand manner. I also expressed my thanks to Mr. C. Jaykumar and Mr. Gautam for the technical support. Thanks to all the participants who have joined us today for this webinar and have updated their knowledge on today's topic. I would like to express my heartfelt thanks. I express my heartfelt thanks to our staff, research scholars, and PG students of Department of Pharmaceutical Chemistry for making this event a grand success, and a special mention to Dr. Muhammad Afzal Azam and Dr. S. Jubi. I also thank all our staff members and students of JSS College of Pharmacy Ooty for their support. Once again, thank each and every individual. Thank you. Over to Dr. Jubi. Ma'am, over to you, ma'am.
Unmute your mic, Jubi. So this presentation was very wonderful, sir, and very informative. I think all our research scholars and faculty got a lot of information, and it will be useful for them, whether for the research work or for the academics. Some new informations are there. So it is. We are very thankful to you, sir, and we'll have the next session again in future shortly, and we will coordinate it, and we'll make it useful, sir.
Thank you.
So thank you very much,
Thank you
sir.
very much.
So thank you so much, sir. Thank you so much. Thank you so much for your
Thank
time.
you, Dr. Zubi.
Yeah. Yes, sir. Thank you, sir.
Okay, thank you. Bye-bye, everyone.
Okay.
Yeah.
Goodbye, sir.
My pleasure,
Goodbye,
sir.
Bye bye, sir.
Thank you.
Thank you.
Bye.
End the session, sir.
Yeah.
Okay, thank you, sir.
Take care.